So thank you for coming to talk.
Sure. Thanks everyone for coming. Yeah, thanks Devin for inviting me to come. So I am a Badger. I got my PhD down at Madison. I actually have a first degree in English from UT Austin. So, you know, you never know where you're going to end up. But I found myself in engineering. And I'm interested in looking at the biomechanics of musculoskeletal tissue. So what we do in our lab is trying to investigate basically what determines the strength of a given tissue, and the tissue I'm interested in typically are things like bones, ligaments, cartilage, so more on the musculoskeletal side, recognizing that there are many biological tissues, right? You could be studying cardio, lungs, arteries, and so on. When we think about the strength of a tissue, and especially in the biological sense, we can consider a couple of different factors that are going to contribute to the strength. So of course, composition, so what it's made of, structure, how are these constituents organized, the environment, so this could be things like cellular environments, or even at a more larger scale, you know, what's happening broader in the body. And then of course, genetics, right, is an important component that it might just be predetermined to kind of have weaker bones or not weaker bones. This is also certainly a factor. And then we have two other external factors that are going to contribute to the strength of those tissues. And this is due to the aging process, typically, unless it's in the injury category, right? So you can injure a tissue depending upon what you do, especially as a child. And then there's disease, of course, again, associated with aging. So we don't focus so much on the environment or genetical components. And our lab is working in two critical areas. One is on shoulder biomechanics. So we're focusing a lot of our efforts on rotator cuff tears and soft tissue mechanics associated with the shoulder. But today I'm going to talk more about bone and specifically the process of what determines actual bone strength as a material. This is motivated a lot by the incidence of hip fractures that are going on. So it was estimated, this is now quite old, but it seems to be on track, that because of the growing population, so there's a lot of older people, and the population in general is getting larger. About 8 million hip fractures will occur by 2050 per year. And it's still the case that about 25%, so 1 in 4 people after there is a hip fracture will die within a year. And this is due to a variety of causes. A lot of it actually has to do with poor healing. So it's not necessarily the fracture itself, but it's the recovery process, it's infection, lack of good blood flow to kind of get the process, the actual site healed. And then still there's about half who have a hip fracture will have some sort of permanent disability. Again, this is skewed towards an older population. So here's an X-ray radiograph of that hip fracture of the proximal femur, which is one of the bones we're going to talk a lot about today. So I'm not a doctor, but I am an engineer, and so I try and understand essentially what's going to make our bones stronger, or what can we measure from that actually tell us if a bone is susceptible to fracture, right? One of the things that we know about bone and what makes it kind of an interesting material to study is that it's an optimized material. So bone is alive, and it's actually constantly responding to mechanical forces. One of the primary mechanical forces that it responds to are our muscles and the ground reaction force. So our muscles are attached to our bones, right, and they pull on our bones. And then, of course, as we do many tasks, we have external forces acting on our bodies. And we can start to look at bone and we see that it has multiple components to it. And if we start to look a little bit closer, we can see that it's actually quite heterogeneous, even just at the structural level. So here we have this spongy sort of bone on the inside that's called trabecular bone. And on the outside of our bones is this harder bone called cortical bone. And if we look at just a piece of cortical bone, what you'll actually find are basically a set of ring-like structures called osteons that are embedded in this other matrix material. So at this scale, if we take a little piece of cortical bone out and put it under a microscope, you would see these sort of tree-like structures. And then if you kind of pulled out from the middle, you would have these series of concentric, basically sheets. So now it's kind of like looking like plywood, right? If you've ever seen multi-layered plywood, kind of comes in these sheets. And what's interesting about plywood, what makes it a pretty strong material, is that oftentimes those fibers are going to be oriented sort of perpendicular to each other. You'd never put plywood sort of in the same grain orientation because then it's not very strong, right? And bone sort of has a similar structure. So if we take a look then and pull out one of those sheets of bone, we can see that they're actually basically made of collagen fibers. And collagen itself is actually quite pliable. And those collagen fibers are then oriented with these gaps. And within the gaps are hydroxyapatite crystals. These crystals, the mineral crystals, is what a lot of people focus on in terms of bone strength. And so when we talk about mineral density, we're talking about how many of these hydroxyapatite crystals there are within this matrix. So hopefully this gives us a definition for some of the vocabulary that I'll be using today. What I really want to point out, though, is that it's obviously multi-scale, and what we don't really know yet or have a good feeling for is at which level is strength globally. If you fall over, at which level do things really start to break down? And some will argue that it's happening at multiple levels and propagates up and down. But we're trying to understand this system of materials and essentially, more importantly, understanding how our muscle forces can actually drive some of this structure and mineral composition that we see. So in order to look at whether or not bone actually responds to muscle forces, there's a lot of different ways we can do this. And here we're just going to look at this whole bone scale. So we did a study on baseball pitchers to actually look at the effect of mechanical loading on bone. So baseball pitchers are a really good model to understand what's happening biologically to our bones in response to mechanical forces due to muscles, because it's a very fast activity. It's incredibly high loads. So a lot of soft tissue injuries will happen in baseball players. And what's nice about baseball players, like other unilateral sports, is that it's unilateral. And that means most baseball pitchers will just pitch with one arm. The other arm, they'll still strength train it, but it's never going to see the same pitching. There are very few pitchers that will pitch on both sides, especially at the major league scale. Tennis players kind of have the same thing. So what that means is that if you're studying these people, they basically serve as their own internal control, right? You can compare the pitching arm to the non-pitching arm and then see what happens over time. And that's what we did. So these are reconstructions of CT scans, which are sort of like three-dimensional radiographs of baseball pitchers. And on the left, you have the non-throwing arm. And on the right, you have the throwing arm. So from the outside, it may be clear that there are some differences, but certainly if we start taking cross-sections of the bone, you can see some pretty big differences in terms of the adaptation of the bone in response just to pitching, right? So starting here at the bottom and then moving all the way up, you can see that the bone in general is bigger, right, in diameter, for example. The wall thickness of the bone within this hollow, the inner part, there's marrow on the inside, but essentially it's mostly that cortical shell. We're not looking really at a lot of trabecular bone in this case, that spongy stuff. But clearly there's a big difference in terms of the wall thickness. So this bone, just because it's been exposed to these really high forces and inertial forces associated with pitching and the training of pitching, has adapted in response to that. If you run a finite element model, so we did this where we computed the muscle forces associated with pitching. I'll go through the process of how we do that in just a minute. There's no surprise, right? The pitching arm is basically stronger in response to the muscle forces compared to the non-pitching arm. So if you applied those same muscle forces associated with pitching to the non-pitching arm, you might break it, right? But that bone, it's adapting over time. So it's been adapting in response to that, which is why it doesn't break. Now, the question is, what happens after you stop pitching, right? And whether or not adaptation that occurs in response to these muscle forces actually stays lifelong. This would be a useful thing in terms of treating bone disease, because what that would mean is if you do exercise when you're young, that can actually change your bones so that they're stronger. If you stopped doing the exercise, there's a lot of people who would think, and there is some evidence to suggest that you would lose a lot of those benefits. So we went and tracked down baseball pitchers at different stages. And so on the very right-hand side here, this is now the mineral density map and structural map of the one cross-section across a bunch of baseball pitchers. So here at the bottom, we have the active thrower. So this is essentially one of these slices over here. And you can see, again, the throwing arm is much bigger. And now this next series up over here is someone who hasn't pitched in less than 10 years. Like within the last 10 years, they've pitched, but or it's been within 10 years since the last time they pitched. And essentially, there's not many side-to-side differences yet. So the adaptation has stayed, even though they haven't pitched in 10 years. And now the next group is 10 to 19 years. And you can see that now things are starting to approach closer to the non-throwing to throwing arm. They're starting to look more and more similar. And as you go all the way up, this is one example slide of someone who has not pitched in 50 years. And what you can see is that the wall thickness has essentially returned back to the non-throwing arm size. What is different, however, is the fact that the throwing arm stays bigger. So the outer diameter of these bones is still larger, and they don't actually resorb and shrink down, just the wall thickness shrinks back to the non-throwing thickness level. A bigger pipe is still a stronger pipe, even if the wall thickness, right, is small. If you can still get that pipe to grow big in terms of just its outer diameter, it's still going to be stronger. And what this means is that the adage of use it or lose it might not actually be true for bone for all geometries. The mineral density levels have returned back to normal, but the actual geometry, the geometric adaptation of this bone has stayed even after 50 years. What does that mean? That means that perhaps there are possible exercise interventions that could be done when we're younger, when we can actually push our bones to adapt in a way that would actually make them stronger later in life. So this was a good model system to show that muscle force, one, does actually change bone geometry and therefore strength. And two, that those adaptive processes can actually stay lifelong, so it might be worth it to develop an exercise intervention. Why do we look at exercise? Currently, right now, one, usually the problem related to osteoporosis, which is a disease of bone quality, let's say, is first of all usually not diagnosed until you actually have a problem, right? So not many of us are going to go in and get a bone biopsy or some clinical scan that can tell us whether your bones are weak or not. It usually happens much later in life. And at that point, the pharmaceutical treatments that will actually try and change the cellular activity associated with the adaptive process of bone are again, often a little bit too late. And so we want to explore the ways that exercise throughout life could be actually beneficial for bone health, but we still have to answer a few questions to do that. One, can we actually design a muscle-informed exercise program? Right now, if you ask a physical therapist or even a bone biologist or bone endocrinologist, for example, they'll basically say, you might have heard, if you walk, if you jump, those kinds of activities are going to be good for your bones. Astronauts go up into space and they have no ground reaction force because there's no gravity. They experience osteoporosis at the same scale that a 65 postmenopausal woman would, right? So there's a lot of evidence to suggest that the ground reaction force is important, but we still don't know which specific tasks. There's lots of ways to generate a ground reaction force and therefore muscle forces. So we don't understand exactly what we would need to do to optimize bone health. And we wanted to ask the question, or answer the question, how do different tasks then load the proximal femur, this bone, which is the one that's broken in that X-ray, and what muscle or muscle groups are responsible for those mechanical loading conditions. Currently, and I think this will probably be forever the case, not many people will let you put a strain gauge in their bones when they're alive. Some people have done this. They have on themselves, so researchers have done this in vivo. But pretty much one of the only ways that we can do this is through computational means. And so there are a set of computational models that can be used that can predict, right, like a finite element model, for example, what the strength of bone is in response to different muscle forces. But first we have to know what the muscle forces are. To do this, we can use rigid body musculoskeletal models. I'm not sure if anyone has heard of OpenSim or anything like that. No. But you can think of kind of a rigid body set of linkages, right? Your bones are kind of like segments that are connected either by pin joints or ball and socket joints, depending upon which segment you're looking at. And they're actuated by muscles, essentially, right? So your muscles act like little motors, and you can solve this system of equations for sum of forces equals ma, or sum of moments equals I alpha. So you need to get the accelerations and the linear and rotational accelerations of the segments, and you can essentially solve for what the muscle forces are. In order to do that, you would bring someone into a gait laboratory where you collect motion capture data. So you put the markers on kind of like they do in the movies or for video games. And you're essentially measuring the segments of the body. And then again, you have people walk over a force plate or do whatever it is over a force plate, and you measure the external forces. And then you set up your system of equations and solve. I'm not going to go through the details of that, but you'll just have to trust me for round that. That's what we did. So we brought in healthy postmenopausal women. We took a CT scan of their femur, and then we can use that CT scan data to reconstruct a three-dimensional model, right? So it's essentially kind of pulling out from the data of the bone, converted into a solid model, which gets converted into a finite element model. So now we have a finite element model of their bone. Bring them into the gait lab. We had them do a couple of different trials. We had them walk a little faster, go upstairs, go downstairs, and inside the stairs were force plates that would measure the ground reaction force. And then we had them jump in place. And we go through this series of computational models that basically get us a finite element model that has muscle and hip forces based on what that person did during that task. So here's an example of the motion capture data. So here she is jumping, and then here's the musculoskeletal model jumping, right? So it's pretty reasonable in terms of the behavior. And then we can build up our finite element model, and using the muscle force predictions from the computational model up above, we can apply them to the FE model, and now we get a quasi-static but dynamic-based trajectory of the muscle force vectors on their bones. So now this is at least a non-invasive way to get estimates of what's actually happening to the bones, right? And we can then calculate a strain value that's a scalar using the strain energy function, and take our models and split them into different portions to see, do any of these activities actually load the bone more? The fundamental premise behind this is that you have, in order to get adaptation, so for those baseball players to get their bones to adapt, they have to do something more than their bones normally see. So as you grow throughout the normal process of aging, your bones are going to grow and sort of scale up normally based on whatever you do, right? Drink water, all the things you would normally do. But if suddenly you do this one activity a lot more, that's what caused that adaptation in the baseball pitchers. In the case of the lower extremities, our suggestion is that the thing that our bones are already adapted for is walking, right? That's the activity that we do the most every single day. That's common to almost all people. So we need to generate strains higher than the strains generated during walking if we actually want to induce an adaptive response. And so this is the way that we can estimate what the strains are during walking and then what the strains are during different tasks. So we broke the finite element model up into a couple of different pieces, and then for each section corresponding to these areas where the bone is likely to break, we calculate the strain over time. So what did we find? So on the left-hand side is the strain in the middle portion of the neck, so that area that's sort of prone to fracture here. And then here in this column is going to be the strain down here, and it's called the subtrochanter, and it's basically where the muscles actually attach to your bones. There are no muscle insertions here. All the muscles sort of wrap up and around and attach to the pelvis. This is strain on the y-axis, and on the x-axis is the stance time. So stance time in gait biomechanics is essentially the time that your foot is on the ground. So during walking, as you walk, 0 would be the moment your heel hits the ground, your body progresses over your foot, and then you push off, right? And as soon as you're in the swing phase, the ground reaction force is 0, and there's not a whole lot of mechanics going on. Some would argue with me about that, but for our purposes, we're just going to look at the time that the foot is on the ground, and so that represents 0 to 100. What you can see is where the muscle force is directly attached to the bone. Of course, we see really high strains, right? That's exactly where the muscles are. The blue line is walking, and the red line is fast walking. What we were happy to see is that if bone is adapting to where the forces are higher, in the subtrochanteric region down here at the bottom, this is the area where the cortical bone, the outer bone, is actually thicker than up here in the neck where it's thinner, right? So there's no need for thicker bone up here because the strains are lower there, right? We need thicker bone where the strains are actually higher and where the muscles are attaching. So this is the data for walking and fast walking, and essentially there was no difference. And then if we look at stair usage, we see similar patterns, so a slightly different pattern in terms of temporal results. But stairs up and stairs down, essentially the same. It's higher where the muscles are, lower in the neck. And then this is the jumping in place data that I just showed you. What was disappointing originally was that it seems like no matter what you do, the strains are the same everywhere. So there's no difference basically between the peak strains for all of these tasks. And we thought, how is this possible? How is it that you would jump, right, and then not see higher forces on your bones during jumping? There's a couple of different reasons, and the same would be true for stair climbing. In the case of jumping, as you saw in that video, she jumped with 2 legs. So it wasn't a single leg jump. It was 2 legs sharing all the force. So essentially half the load gets distributed between both legs. I explained that we're looking at the stance phase for walking and stair climbing. And essentially during these tasks, the other leg is in the air. So as you're walking, you hit the ground. And as soon as you start trying to propel your body forward, your other foot is in the air, and now all the weight is down on the foot that's on the ground until you come through and then touch down with the other leg. But still, how is it that walking and stair climbing, which are 2 very different tasks, and stair descent could create the same strains? And it essentially turned out to be just a problem of over-averaging. So these are the values if we average across that whole section. But if I pull out that middle neck of the bone, right, and now I split it up into quadrants and kind of look at different pieces of the pie within this same region, we actually see a very heterogeneous strain distribution. So essentially before we were averaging across the whole cross section, but the strain response is actually really spatially heterogeneous. So it's mixed up everywhere, and you get kind of this patchwork pattern. These are averages across the 25 subjects. So it's not as continuous as we expected. But now we can see that there are certain areas in the bone that are in fact higher than the walking strains. And if we actually look at the numbers, and again, this is the area that's prone to fracture, what we want is to find an activity that generates strains higher than walking, that first bar. And you can see that for different areas, different activities will do that. It turns out jumping actually does induce higher strains, but only in that little pocket of this portion of the bone. Whereas in the other regions, stair climbing tended to do more. So this was good news. So it seems that stair climbing and jumping are in fact going to do something more to your bones. And the question is, which muscles are actually driving that? So we went back to the finite element model and we applied individually each muscle and its joint reaction force to the FE to understand how much of this strain was coming from glute max, iliosoas, and so on. In the case of the hip, there are 23 muscles that span the hip, but the big ones that are driving the strain are your glutes, your vasti, and iliosoas, which is a muscle that connects the hip to the proximal femur. It's a bit clearer to see it in this graph. So again, walking is the baseline. So when you walk in this portion of the bone, most of the strain is coming from iliosoas. But as soon as you start stair climbing or going downstairs or jumping in place, you suddenly activate glute max. And all of this comes from the fact that in order to do that, these tasks require more hip flexion. So climbing the stairs, right, as opposed to walking means you've got to get your foot up and over onto the step. As soon as that activation, in order to do that task, you basically need to start using your glutes, especially as you get up and then raise your whole center of mass up and over to the top of the step. The same thing happens when you descend stairs and then jumping, as you can see in this case for these ladies, they wanted to, we didn't have them in harnesses when they were jumping in place. So in order to stabilize their jump, unlike what you and I might jump like, they basically really flex their legs, right? They're basically trying to absorb the energy associated with the impact. And so as they jump, in order to try and accelerate their bodies up and then absorb the shock of the landing, they're basically going to bend down quite a lot, right? As opposed to a straight-legged jump, which might send a jolt through the body. That might be good for bones, but if you're 65 and you're already postmenopausal, that might not be so good. So this is what we've learned, right? We've learned that muscles, and we have a means now for identifying which specific muscles can actually create an adaptive response in bone. And what we want to know now is how does this process actually happen at another scale level? So we think we can generate some sort of pattern here, but one of the questions we have is, what is our target for these exercise programs? What are we actually trying to do? Are we trying to increase the thickness of the bone? Do we want to decrease porosity? So you might have heard of osteoporosis, right? Which is, again, I think I said, the disease associated with bone fragility. And if we look at the osteon level, I'll explain where the pores in osteoporosis comes from. So here I'm taking a cross section. So these are sort of the tree ring-like structures within the osteons. And within every osteon, we have to have pores. Our bones aren't designed to be completely solid because they need blood vessels to supply nutrients to stay alive. Those blood vessels run through the center of those tree rings through what's called a Haversian canal. And it's in the Haversian canal that a lot of remodeling, which is the process of bone turning over, happens in order to, for the cellular activity to take place. Now, osteoporosis happens when those canals get really big, essentially. And the processes behind this are still not very well understood. So if we want an exercise intervention, we still need to know what our target is, right? Some level of porosity is needed, but how much is too much? And what are the drivers behind that is something that's still being investigated. So we started working with some bone biologists to look at this, and I'll just take a minute to explain what the pore remodeling pathways look like. So you now know that bone is dynamic and it's constantly growing, right, in response to mechanical forces. And if we start at the top, this is time moving down. And if you looked at what was happening in bone, there's essentially a couple of phases. In order for bone to grow and change and adapt, a couple of different things have to happen after skeletal maturity. First, your bone is set, and the first thing that happens is the osteoclasts come in. So osteoclasts are just bone-eating cells, so clasts, cleavers, that's the way I think of it. They will come in, and that's what's shown at the top, and they basically act like little tunnel drillers, right? They're kind of like English Channel tunnels in our bones. And they come in and they kind of clean the surface off. Why they do this and how they do this is still being widely explored, but this phenomenon is well documented, that there's a sort of cutting cone. These cells act like little drills. After that, there's sort of a reversal phase, and they say, hey, I'm done. I'm ready for new bone to fill in. For us to stay healthy, basically what needs to happen is whatever they removed needs to get filled in, right? It's kind of like repaving a road. You fill it back in, and these osteoblast cells come in, and they actually build bone. So the cutters, the cleavers, the osteoclasts start first. They signal to the osteoblasts that they're done, and then the osteoblasts come in and build more bone. And after a while, that process will finish, and you'll get the formation of one of these ring-like structures. And this process happens only on the surface. It's never going to happen in the middle of the bone matrix. It's a surface-based remodeling phenomenon. But there's a couple of problems associated with aging. And one, what we think might be happening is that whether or not that process happens in two pores that are really close together, and it might be one of the reasons that suddenly pores can suddenly appear very large and then lead to what's documented, right, as osteoporosis. So here I'm explaining, again, the same phase, but now I have two independent pores. So this pore is a cutting-cone pore generated. I can't see my, oh, here it is, right here. So here there are these two pores. This one is starting the digging process. This one has started the digging process within the osteon. And there are a couple of different pathways that this can happen. And essentially, if there's another pore really close when you're still digging and they're both digging, it's possible they're going to dig into each other, right? And then suddenly you get this really large pore. What's supposed to happen, right, is that then still they'll say they're done. They're going to start, stop the digging, start the building, and then complete the formation process. But if they've coalesced or merged, you might end up with a bigger hole. And so we wanted to know whether or not this process associated with whether or not these pores coalesce or independently existing is actually driving the change in the mechanical strain in the bone, and whether or not this process actually completes. So to do this, we acquired human bone samples from people undergoing a jaw reconstruction. So if you have a facial injury that requires bone grafts, essentially, they'll take bone out of your fibula, which is the thin little bone in your lower leg. So I guess it doesn't see a lot of load. I'm not sure what they put in its place, but they essentially take out a little piece, and then whatever was left over after this surgery, they gave it to us so that we could analyze these bone samples. The bone biologist went and looked at the histological measurements, and they basically figured out which cells were acting in each of these different slices of bone. And then we did some structural analyses based on those slices. So we counted up all the pores, all the holes. We quantified their size, so how big they were, and how circular. So, mechanically speaking, an irregular object might induce higher strains and strain concentrations than, a circle is essentially an optimized shape. And then we figured out where they were relative to the edges of the bone, and then took a subsample and then ran a finite element model and compressed them, and then looked at the strain just around the bones, around the pores. So here are some examples of some of the results. You can see sometimes the pores are quite large, but there are other bone samples that actually have many small pores, but not necessarily huge, like this one over here. So the typical question that people would ask is, okay, so are these big pores really the problem? Is that where all the strain is coming from? And is that what's contributing to the fragility of bone? But we decided to turn the question around and first try and stratify the pores. Which one of these pores are bad pores? Just looking mechanically at the strain energy around the pores. So if you took a given sample and then you just plotted them on a statistical box plot, anything that falls as an outlier that has abnormally high strain, we said, okay, there's something weird about these pores. We don't know what it is, but they have high strain energy relative to the other pores within the sample. And when we look at the results, we can see that per sample, we have a set of pores that were outside of that statistical distribution. And then by categorizing them into these two categories, we tried to determine what was different about these two sets of data. In terms of size, no surprise, the high strain energy pores were the bigger ones, right? So we didn't pre-distribute them by size. We figured out what was unique about the high strain energy pores structurally. So they were bigger, they were more irregular, so their circularity, the high strain energy pores on the right-hand side tended to be more irregular, so they were these blobby shapes. And they were more preferentially located closer to the endosteal side, which is the side on the inside of bone. So now from the histological measurements, whether or not they were pores associated with the erosive phase, so the bone-eating phase or not, we found that the high strain energy pores were mostly pores that were being eroded from within the existing canal compared to the low strain energy pores. So that's what we are calling type 2, the ones that are sort of eating from within. And then whether or not they actually broke out of their Haversian canal and then through the lamellar wall and coalesced was next investigated. And we found that, again, the high strain energy pores tended to be the ones that were a result of these coalescing pores. This seems like a pretty obvious result, right? Like of course, big pores that merge together are probably problematic. But before this data, this had really never been shown conclusively and with the actual cellular activity to support it. And then whether or not they actually complete this process was also investigated. So theoretically, these should be kind of quiet, happy pores. And what we found is that, again, the high strain energy pores were essentially, they're stuck in the erosive phase. So they never complete the full remodeling pathway. And what that means is that essentially you've scraped out a bunch of bone, but it never got to the filling in process. So cellularly, something is happening. These cells aren't communicating properly. And this is really the next thing to be investigated. So we want to improve on these models by incorporating their morphological characteristics, include weight-bearing bones. So the fibula is not typically a weight-bearing bone, so maybe it's not optimized for strength, and then validating the models. I want to quickly, if I can, in the last 5 minutes here, get to our last topic on what is the actual target. So again, we still don't know really what what level of porosity is normal, what thickness of bone is normal. I've shown you some information that connects the cellular activity to the structural response, right? The result of that cell activity. But we're still struggling to understand what would be missing from our models diagnostically so that if we took a scan of someone to know whether or not their bone is healthy or not. And so to start answering this question, we've actually turned to looking at animal models and starting to look at how does bone just develop normally, right? So from a kid to an adult, what decisions is bone making as you're growing that says, I need this number of pores, I'm gonna need this many blood cells or vascular canals and so on. And then specifically, as you grow, your muscles are growing stronger, the joint forces are getting bigger, and how is bone scaling up to be an optimal structure? So I'm gonna skip right to this slide. This idea of how do objects scale to get bigger is an old idea. It started with Galileo. I'm sure someone before him thought of it, but he wrote it down. And what he observed was that if you look at, for example, the bone of an elephant compared to the bone of a human, elephants are, I don't know how many times bigger than we are, but their bones aren't proportionally bigger, right? So if we think about just the dimensional analysis process, right? Surface area, as you know, is a function of length squared, volume, length cubed. And then we can relate mass to volume just through density, right? So mass equals density times volume. So using that last equation, we can say, right, just take this to the third on both sides. Length is basically proportional to mass to the 1/3 power. And if we square length, we get area proportional to mass to the 2/3 power. So this is a way to relate something that's increasing in mass to an idealized cube, right? In this case of dimensional metric. So J here is the moment of inertia. I'm not sure how many people are good with that, but that's essentially an indicator of bending strength, you could say, right? Area would be a good indicator of compressive strength, force over area. So if we just did, again, a simple dimensional analysis and looked at stress as force over area, we would find that stress is proportional to density times gravity times length. Now, if you double the length of everything, so you just isometrically scale, you just pull out, right, click with your mouse and then drag it bigger and double the length of everything. Area goes to 4L squared, volume to 8L cubed, and mass, so on. And now if we did the stress calculation, we would see that if you just scaled this object up, the stress actually doubles, right? This would not be good, right? If all our bones did was just multiply all their dimensions by the same constant, it's actually now under more stress than when it was before. And biologically, this has been questioned whether or not this is really the true mechanism that would make sense for growth. So if you took, I don't know if this is actually true, but I found this nice picture, right? Little lizards, right, and just scaled them up, their bones theoretically would be under more stress as they get bigger. In humans, this is definitely not the case, right? When you're a baby, your femur is about this long. As a skeletally mature adult, it's probably maybe ×5 in size, right? Your head is certainly not 5 times bigger than it was as a baby. So different parts of our bodies are scaling at different rates as we're growing. That process of deviating from just isometric scaling is called allometric growth. And if we assume that, of course, density, mass, volume are still related by the 3rd power, in order to have a constant state of stress, these are the values that the area and the moment of inertia would need to scale to if we want stress to stay constant, or at least not increasing during growth. So we took rats at different ages, and we followed them actually over time. So the same rat was brought into a lab. We built a gate laboratory for the rats, so it's essentially like a glorified aquarium. There's a force plate underneath. We put little markers, we just Sharpied little markers onto their bones on the outside, just like we did in the postmenopausal women, and then track their gait over time from 8 weeks weekly to 12, 14, and then endpoint 20 weeks. And then we also collected micro CT data of these rats as they were growing, as they were alive. Here's the increase in the peak ground reaction force. So of course, ground reaction force and mass are sort of synonymous. So these are skeletally growing rats. They haven't quite reached maturity, but they're certainly getting close. And then we looked at the cross-sectional properties of these rats. So per rat, we had about 1,000 images per bone, per time point. And this is the cross-sectional area in one rat across time. So the blue inner line here represents time point 1, and then it's slowly scooching out, right, as this rat is growing. We've normalized them so their center line is at the midline. And then this is cross-sectional area in every single slice within this data. And you can see that on the bottom side, at the end, doesn't seem to be a whole lot happening, but there seems to be a whole lot more happening on the right-hand side. And so we started analyzing what was going on in these 3 different regions. We can plot cross-sectional area as a function of body mass on a log scale, and we'll essentially be able to come up with those exponents that I showed you before about growth. So in terms of compressive strength, cross-sectional area, none of the cross-sectional areas actually achieved an isometric or allometric scaling rate. But bending strength, what we found is that in the diaphysis, in the very middle of the bone, the rat is growing almost exactly according to a static strain similarity value, which was pretty surprising that we got this number. And so what explains that? It's the fact that essentially this is suggesting to us that these bones are under more bending loads compared to compressive loads. If it's optimizing in response to the mechanical forces that it sees, then this is suggesting that this bone actually doesn't see that much compression, so it doesn't have to scale accordingly. What it does suggest is that it's actually seeing more bending, and more bending is happening right in the middle, right? The purple line is the middle line. These other 2 don't scale isometrically or allometrically, but this one does. If you take a pipe and you bend it like this, the most deflection is going to be right in the middle, right? And so this data sort of supports this idea that this bone is actually subjected to more bending forces. If we look at the actual joint angle, so we can look at the kinematics and see what's happening. So unlike you or I, where we stand with erect postures and all of our forces coming straight down in compression, essentially on our bones when we're standing completely still, rats don't walk that way, right? They're like crouched down, and they have incredibly high flexion angles. So 180 is completely extended. So the lower the number here is actually more flexed. But essentially, they become more flexed as they grow, and then they start walking the same. And so finally, the mineral response in the bone is also following a similar pattern. So not only in response to that bending load, we can see that there are changes in the mineral density, again, peaking at around 3 or 4 months, 4 weeks after our first time point. So what are our next steps? This is my last slide. We're looking at doing this in animal models, so there are some limitations associated with rats. So we're tracking horses over time now. So these are foals that are in our barn there at UIUC, and then looking at a couple of ways that we can actually measure the 3D micromechanics of these structures. So I'm going to wrap it up with that. But I do want to thank all the members of my lab, our funding sources, and again, Devin, for giving me the chance to talk to you about bone and how it scales. I'll be really happy to answer any questions you may have.
Thank you very much.